So the process, like I told you, the process is very simple. We have the DNA that goes into transcription to give us the RNA that can then move out of the nucleus and then be translated to give us the protein. And on the eukaryotes, or even on eukaryotes, if you remember, this translation process, the proteins enter the rough endoplasmic reticulum, which means the translation process should take place on the rough endoplasmic reticulum, right? And so the RNAs should all gather together on the rough endoplasmic reticulum. And do you remember the rough endoplasmic reticulum was special because it had little blobs of ribosomes on it. Those ribosomes are in fact the ribosomal RNA or the rRNA combined with protein to form those blobs of ribosomes with the heavy chain and the light chain, okay? So that's in fact what it is. So it's been 45 minutes already. I think, should we do 10 more minutes or are you guys tired? Do you guys have the energy to learn for 10 more minutes? Yeah, it's fine for me.
Let me ask my ATP. No.
Yeah, you can ask
Yeah,
your ATP. That's yeah, very nice. Okay, so let's just do it quickly, okay? So okay, so now the first step, like I told you, is the DNA splits open because we need to access it. This opening will then expose the strands. One of those strands is used to make this RNA. This RNA is made from one of these strands, as you can see, because this is used making something called a polymerase, a specific, that the previous one was a DNA polymerase, this is an RNA polymerase. Again, because it's a polymerase enzyme, it's going to work from the 3' strand to the 5' strand of the DNA, right? So this is why it's choosing this strand and working on this one to give us this transcript RNA, which is an mRNA. And then this mRNA is packed to protect it. It is given a head and a tail. It's not shown here. I'll show you the details of it later. For now, all you do is appreciate these hydrogen bonds being broken, the RNA being formed from one of these strands. It's not like DNA replication. Only one strand is being involved to give us an mRNA or a messenger. The small m stands for a messenger RNA. And the last step is obviously translation, which is forming the protein. So transcription occurs within the nucleus, gives us DNA to RNA. When this RNA is giving a polypeptide, that step is called translation. Okay. Very easy. Again, structure of the mRNA. You can appreciate the phosphodiester bond over here. There's this question Tanya sent me about identifying nucleotides. So what is a nucleotide? A nucleotide is just a, why am I calling this a phosphodiester bond? Because this is not one nucleotide. These are two nucleotides, okay? Appreciate this one, the ribose, the base, and then the phosphate. The other stuff is not part of this nucleotide. This nucleotide is limited to these three things. Just simple biochemistry. I've taught you this in the first lecture. So please don't disappoint me and try to remember these things, or at least go back and study them. Okay. And then you know the 4 bases. If this was a DNA, it would be uracil, right? mRNA, the M in mRNA starts for messenger. Why? Because it's carrying the message of the code, the gene. Okay, very easy. Again, very simple stuff. You don't need to cram it. All you need to do is try to understand it. If we are clear up till now, then we move on to this scary little molecule called the tRNA. Okay, and why must it be called the tRNA? Just imagine, just imagine for 1 second. I keep telling you it's all concepts. Why is this one called a tRNA? And the answer is very simple. I've already told you why, but let's see what you guys comprehend. So I'm saying mRNA is messenger RNA, rRNA is ribosomal RNA.
Transport?
Okay, so transport, but in this case, it's a basically, transfer? Yeah, it's transport, translation, transfer, whatever you'd like to call. So yes, you guys are correct. So what is it doing? It's transferring or transporting these amino acids. Okay, see, you guys are getting smarter, man. I don't think I need to teach you guys anymore. You guys need to teach me now. Is that the case? Yeah.
Yes, ma'am.
That's great.
No, of course not.
Okay, so now how is this one different from an RNA? I just showed you an RNA. It's a single chain floating around like a snake, ready to go do some work. Okay, how is this one different? It is a single chain as well. The only thing that's different is that that single chain has hydrogen bondings that lead to all of these infoldings upon themselves. You can appreciate complementary base pairing among itself, which is why it gives these three outpouchings. And what are these outpouchings? In fact, they are the single nucleotides being exposed outside that have not been involved in any kind of hydrogen bonding on the inside because there was nothing left. All of these hydrogen bonded amongst themselves on all four ends, and these three poked out because they had no other pair to hydrogen bond with. And that is important for a reason. Okay, I tell you the reason just in a while. The second thing that's important in the structure of a tRNA is to recognize that this area will always be ACC, an adenine cytosine cytosine. Whenever, for instance, your DNA makes two RNAs, one of them has ACU and the other one has ACC. Now, how do you know the fate of these? This one is going to go make an mRNA and this is going to make a tRNA. How do I say that this one's going to make a tRNA? How do I say that? Because I looked at the first few chains. This one had ACC and this one had ACU. And ACC at the 3' end always gives us a tRNA. See, it's telling you these three bases are always the same. They always read as adenine, cytosine, cytosine. That's how, because it's not magic. It's some logic right there. It's never magic, trust me. Even if somebody's telling you it's magic, it's bullshit. There's nothing like magic.
So UGG can also be like tRNA then?
No. It can't, because you're imagining base pairing. RNAs, I told you, exist as single strands. The DNAs love to exist as double strands.
Oh, okay.
RNAs love to exist as single strands. So ACC will always be for a tRNA. It can be different, but if it is different, then it won't have these internal base pairings present to give you this weird cloverleaf structure with three of these poking out. Okay, one last thing, and even look at the look at this. Oh, why does it have to change? Okay, look at this end. Look, just look at this end for me, will you? Can you see that the 5' end is totally booked? The 5' end is totally complementary bonded, but the 3' end has these three free. So that is also one of the properties. Why do we need these three free? Because the amino acid comes in and attaches here. For some reason, some bonds take place that when the amino acid comes in, it hops onto this part, occupies this part. Okay? Is that clear?
Yes, sure.
Then if this point is clear that an amino acid is going to come in and hop onto this part, then I have a very simple question to ask you. Will every tRNA carry every amino acid randomly? It sees a amino acid and it's like, oh, this one is really beautiful. I just want to take this one and not the other one. Is that how it happens?
No, teacher, specific codon binds to specific anticodon.
That is an absolutely incorrect answer.
Okay.
Yeah.
Or the anticodons are specific to each amino
Yes, who
acid?
said that? That's beautiful. See, that's the
That's
thing I want
me,
from you
Tanya.
guys. Very nice, Tanya. That's what I want from you guys. You guys are answering stuff based on speculations. You read stuff and you decide, okay, that's what's happening. But in fact, that is not what's happening. What's happening is that you need to know the exact stuff. And because you don't know what a codon or an anticodon is, which is why you gave me that answer. So let's see first. Let's decide what those two words mean, right? So let me give you a simple hack. Let me just tell you something simple. There's a very simple rule. 3 of these base pairs code or give instructions for one amino acid. So if I say this is leucine, then these 3 combined will give me code. What do I mean by code? Have you ever done a computer language? There's this 1, 0, 0, 1, 0. These individually mean nothing, but when a sequence of these in a specific order comes in, they make a special code that you and I cannot read because we're thankfully biology people, but people who are doing computer science can definitely read these, right? They'll tell you immediately this means this and this means that, right? That's how they code. In our language, we code by using these 3. And a pair of 3 will give us one amino acid. So if this pair gives us one amino acid and this is a pair, it gives us another amino acid, and I tell you this is complete for a protein, then how many amino acids does this protein have?
2.
2. Very simple. These will bond and give us a protein, right? So if I tell you a chain of mRNA has 9 nucleotides, how many amino acids does this chain have? It's very simple. If 3 nucleotides give us 1 amino acid, 3 nucleotides give us 1 amino acid, then 9 nucleotides must give us X. And every time this ratio is the same. So all you have to do every time is divide your number of nucleotides by 3. Right? So how many amino acids? The AA stands for amino acids. So how many amino acids do 9 nucleotides give us? Come on, guys. You're too slow.
3.
Thank you. Thank you. Okay. So now this 3 pair is called a codon. Okay. This pair is called a codon. What the part on a tRNA that attaches or is complementary to this pair is an anticodon. And what you need to remember over here, first things first. If this is the mRNA, I ask you for the tRNA. I ask you to give me the base pairing for this on a tRNA. If this is the codon, what will be the anticodon for this amino acid? So this is going to be C. The U will match with the
A.
A. The A will match with the
U.
U. Why not T?
It's an R.
Okay.
RNA.
And now this sequence. So if this sequence is present on the mRNA, this sequence will be present on the tRNA. And because this sequence is the opposite of this sequence, it's called the anticodon. So if you understand this, then one thing is pretty clear. If this is the DNA and the mRNA is the opposite of I use this strand of the DNA, the mRNA is going to be the opposite of this strand of the DNA, right? Because it's supposed to be anti-parallel. Base pairing is anti-parallel, right? Is that clear? So this mRNA and these 3 on the mRNA are a codon. Then the tRNA, these 3 on the tRNA are the anticodon. Why? Because they are the opposite of the mRNA. But they must be similar to the DNA, right?
Yes.
So if he gives you a tRNA only and asks you for the DNA sequence for these 3, will you be able to make them out? They should be the exact similar, right? They should be C, A, and T because DNA does not have U. Is that not so?
Yeah.
Can you use a number of codons to give me anticodons? and then use a number of anticodons to give me DNA? Or can you use a number of DNA to give me a number of anticodons on a tRNA? Because all of this is just stepwise. Isn't it stepwise? Is this clear? Do you understand what I mean? And this is exactly what they're going to do. They're going to give you a sequence of amino acids, and then they're going to ask you, okay, tell us the anticodons for it. Now you should know where are the anticodons. The anticodons are part of the tRNA. So I want the tRNA sequence for these. But in our lists, in the lists that we use, we know that codons code for amino acids. Anticodons do not code for amino acids, codons do so. So in a list, if you're given a list, what you're going to do is you're going to see that amino acid, you're going to decide, okay, for instance, I tell you that glycine has the codon for glycine is AUG, okay? And then I ask you, what's the anticodon? So can you give me the anticodon for glycine? Isn't it going to be the opposite?
Yes.
So what's it going to be?
AU.
What's the anticodon going to be? So it's
UAC.
AUG, so you have UAC, right? And then and in the next part, I asked you what the DNA is going to be. What's the DNA going to be?
T A
Yeah.
T
Yeah.
G.
TAC. No. It's going to be a TAC. It's going to be the exact similar. That's what I'm trying to tell you. If this one, if mRNA is the opposite of DNA and tRNA is the opposite of mRNA, then tRNA is in fact DNA. Okay, let's do this again. Let's do this part again. So if I have a DNA that codes AU, DNAs can't have a U. So this one codes, my screen is acting up. Wow. It codes ATG. What's the mRNA going to code? Guys, what's the mRNA going to code?
AUAC.
Okay, I don't know why my screen is acting up like this. Okay, never mind. Okay, and so what's the tRNA going to code? A U
G.
G. Isn't this and this similar?
Yes, it is.
So in fact, tRNAs and DNAs are the similar. Replaced obviously the T and the U. So is that clear?
Yes.
Yes, teacher.
Okay, that's great. Okay, so now let's see, we have this, and then we have the amino acid attachment area. What decides what amino acid is going to attach? This anticodon. Why do I not call it a codon? Because codons exist only on mRNAs. Anticodons exist on tRNAs. Okay. Which is why we have these three sticking out, because what eventually happens is, this is where I tell you all three of them combine together to form what they form. Look at this tRNA with the free attachment end for 3', and then there's loops. You can look at the diagrams and know their names, a T and a D loop. You can see this anticodon loop hanging out with the anticodon that will pair with the codon. It is going to bring in the amino acid here, and all of this is going to occur. The active site where this is occurring is the ribosome. And what was the ribosome made up of? The third RNA, the rRNA, right? A small subunit here. This is the smaller one and this is the larger one. And as a result, any amino acid that is brought in by tRNAs, for instance, another tRNA, I draw over here, brings in another amino acid. These will bond together to give us a growing polypeptide chain. Okay, So eventually all three of them are acting together. Now I think this much is enough for today. We'll do the details for translation tomorrow. We've done, so it's very easy. You watch the video, it's going to be a 2-minute thing. The rest of it is just examples of translation, so not very difficult. We're essentially done with the topic, so you can go back and read the book. Okay, guys?
Teacher, for tomorrow, can you record the class? Because
Yeah, sure.
I think we cannot able to join.
We won't, yeah.
We won't. So.
Okay, that's fine. I'm going to record it. No worries.
Thank you, Miss.
teacher.
Any questions
Bye.
today?
No,
No,
Miss.
no.
Perfect.
No.
Best of luck. Goodbye.
Goodbye.